I was thinking about how we traditionally solve crimes.
Oh, yeah.
In the old days, a detective walks into a room, snaps a photo of the scene, and they try to figure out what happened from that one frozen moment.
Right. You see the aftermath, but you completely miss the action, the motive, everything that led up to it.
Exactly. And for the longest time, that is exactly how we have treated biology. We take a cell, we freeze it, we sequence it, we get a snapshot.
A single Polaroid.
Yeah. But life isn't a photograph, it's a movie. A stem cell doesn't just sit there. It travels a path to become a neuron or a muscle cell.
Yeah.
And if you miss the frames in between, you miss the entire story.
That is the fundamental frustration of developmental biology. We've been staring at Polaroids trying to understand a motion picture.
Which is why I completely geeked out over this new paper we're discussing today. It's from Science China Life Sciences, published late 2025. A team at the Chinese University of Hong Kong.
And they haven't just built a database. That's the key thing.
No, it's more like a time machine for biologists. It's called TEDD 2.0.
Temporal expression during development database.
Yeah.
And you're right to call it more than a database. A database is like a filing cabinet. You put data in, you take it out.
This is different.
TEDD 2.0 is an active computational engine. It doesn't just list genes, it tracks them through time, across species, and this is the part that blew my mind. It lets you simulate experiments right there in the code.
That's that in-silico aspect we kept reading about.
Exactly. In-silico meaning, in silicon on computer chips rather than in vivo in a living body.
Okay, so here's our roadmap for this deep dive. We're going to unpack this thing layer by layer. First, the sheer scale of the data because it's massive.
It is.
Then we're going to dissect its three, let's call them superpowers. Virtual gene knockout, the cross-species translator, and the time-traveling trajectory map.
It's a perfect structure. Because you can't really understand the tools until you understand the raw material they're working with.
Right. So let's start there with the foundation, TEDD 2.0. We are talking about 15.2 million cells.
An incredible number.
And that number alone is staggering. But it's the variety that really counts here, isn't it?
It is. They've integrated 162 unique time points across 85 different tissue types. But the real headline for me is the species list. Most databases are just human and mouse.
Maybe rat, if you're lucky.
Exactly. TED 2.0 has curated data from 9 species.
And for this 2.0 update, they added 5 new models that really bridge some important gaps. They added the cynomolgus monkey, the marmoset, the rhesus macaque.
The primates are close cousins, which makes sense.
But then they added the domestic pig and the European rabbit.
Which might sound like a sort of a barnyard random selection, but it's actually incredibly strategic.
I thought it might just be about availability, but the paper argues it's all about anatomy.
It is. Think about a mouse heart. It beats, 600 times a minute? It's tiny. If you're studying human heart development, a mouse is a pretty terrible proxy.
But a pig.
A pig's organs are remarkably similar in size and physiological function to a human's.
So for organogenesis, the actual growth of organs, the pig is a much better mirror.
Precisely. And the rabbit, it offers a different advantage. Its embryonic morphology, the actual shape and structure of the embryo as it develops, is distinct from rodents and actually shares some key features with primates.
So essentially, TEDD 2.0 lets researchers triangulate.
Yeah.
If you see a gene doing the same thing in a mouse, a pig, and a rabbit.
Then you can be much more confident that it's a fundamental rule of biology and not just some weird quirk of being a mouse.
It's about building a stronger case with multiple witnesses.
Exactly. And the best part is they've wrapped all this data in a cloud-based interface. It's code-free. You don't need to be a Python wizard to use it. You can just log in and start asking questions.
Okay, let's recap this first part then. We have this massive library of biological movies.
15.2 million frames of film.
From the barnyard to the jungle. Now, let's get into what we can do with it. This brings us to that first superpower. Module 1, the virtual gene knockout, or VKO.
This is the feature that is going to save the most time, the most money, and potentially the most animal lives.
So to set the stage for you listening, in the real world, if I want to know what a gene does, I usually have to break it.
Right.
I breed a mouse that is missing that gene, a knockout mouse, and I wait months, maybe a year to see what happens. It's slow, it's expensive, and frankly, it's pretty crude.
And VKO lets us do that exact deletion mathematically in seconds.
But this is where I need you to help me understand the how. The computer doesn't have cells. It doesn't have proteins. How can it possibly know what will happen if I delete a gene? It feels a little like magic.
It feels like magic, but it's just really, really good math. It relies on a machine learning framework called Chen L. Okay, so here's the logic. The system analyzes all that wild type data, the healthy and normal cells. It looks at all the interactions. It notices that every time gene A is loud, gene B gets quiet. And when gene C turns on, gene D turns off.
So it's mapping the social network of the cell, who talks to whom.
Exactly. It builds a gene regulatory network, a GRN. Once it has that map, you can tell the simulation, okay, now remove gene A.
And it calculates the ripple effect.
It ripples that change through the whole network to predict how every other gene will react based on those established rules.
The paper gave a specific example that really crystallized this for me. They looked at a gene called TREM2 in microglia.
This is a perfect case study because TREM2 is a huge deal in Alzheimer's research.
Oh, wait, I've heard of it.
Microglia are the immune cells of the brain. They're the janitors. And TREM2 is the gene that helps them sense and clean up lipids or fats. If TREM2 is broken, the trash piles up, and that's a hallmark of Alzheimer's.
So the researchers went into the TEDD 2.0 simulator. They took healthy mouse microglia data, and they virtually deleted TREM2. What did the computer predict?
The system identified 89 differentially regulated genes that went haywire because TREM2 was missing.
Now, how does the computer decide what counts as haywire? Because, biology's noisy. Is there like a score?
There is. They use a metric called Kullback-Leibler divergence, or KL divergence.
Which sounds incredibly intense.
It's a concept from information theory. So imagine two probability distributions, two different shapes on a graph. One shape is the gene expression with TREM2. The other shape is without it.
Okay.
KL divergence just measures how much surprise there is when you switch from one to the other. A high KL score means the system is basically screaming, something has changed dramatically here.
Okay, so the algorithm flags these 89 genes with high KL scores. But again, computers can hallucinate. How do we know this isn't just a very fancy guess?
That is the million-dollar question. And to prove it, they compared the virtual results against a real biological experiment where scientists actually bred mice without the TREM2 gene.
And the scorecard?
It was remarkably accurate. 35 of the top virtual predictions matched the real-world biological data exactly.
Can we talk about a specific gene? Because I think that's where the proof is.
Absolutely. The simulation predicted that a gene called LPL lipoprotein lipase would be the second most affected gene.
And in the real world?
In the real world, LPL is an enzyme that breaks down fat, and it is significantly downregulated in TREM2 knockout mice. The computer predicted that relationship purely from the network map of healthy cells.
That is wild. It also mentioned the DAM state.
Right, the disease-associated microglia state. This is a specific profile that microglia switch into when they're fighting plaques. The virtual knockout correctly flagged genes like ApoE, CTSD, and TYROP, all hallmarks of this disease state.
So just to recap this section, because it's huge.
Yeah.
We took data from a healthy mouse.
Right.
We asked a computer, what happens if we break TREM2?
Yep.
The computer used a network map to predict that the fat-cleaning genes like LPL would fail and the Alzheimer's genes like ApoE would flare up, and real life confirmed it.
You nailed it. And think about the implication. You can now screen thousands of genes virtually to find the one that might matter before you ever spend $1 on a lab experiment.
It filters the haystack so you can find the needle. But that leads to the next problem. We are still talking about mice. And as we said earlier, mice aren't humans. If I find a cure in a mouse simulation, does it actually matter for a human patient?
This is the infamous translational gap. We have, cured cancer in mice thousands of times, but those cures often fail in humans because our gene networks are just slightly different.
So TEDD 2.0 has a tool for this too, Module 2, cross-species integration.
Correct. And they address this with a pipeline they call BENGAL,
which stands for
Benchmarking Strategies for Cross-Species Integration. It's a systematic way to solve the apples to oranges problem. You see, human data and mouse data come from different labs, different machines. We call those batch effects.
So if you just dump them on a graph, they won't line up.
Not at all. BENGAL is the translator.
A very rigorous translator, it sounds like.
It is. They didn't just pick one method. They ran a tournament. They tested 24 different strategies, combining 8 integration algorithms with 3 types of homology mapping.
Okay, let's pause on homology. That just means genes that share a common ancestor, right?
Yes, but they tested different levels of strictness, one-to-one orthologs, which are direct equivalents.
The strictest.
Then higher expression, which includes similar families of genes, and strong homology, or SH, which focuses on high-confidence matches.
And the winner was?
A method called harmony combined with that strong homology. It was the Goldilocks zone. It scored highest on species mixing, so the human and mouse cells blended together on the chart. But it also maintained biology conservation, meaning neurons still clustered with neurons.
So they proved they could overlay the maps. Did they check our friend TREM2 on this combined map?
They did. They integrated adult brain tissue from humans and mice, generated these UM-app plots. Those are the scatter plots where every dot is a cell. And they looked at where TREM2 lit up. It was a match. TREM2 expression was high in the microglia of both humans and mice. They even used a statistical test, a Wilcoxon-Rank sum test, to prove that TREM2 was not a species-specific marker.
So in plain English, the computer couldn't tell the difference between the human TREM2 activity and the mouse TREM2 activity.
Exactly, which validates the mouse as a model for this specific gene. It tells the researcher, yes, for this, the mouse is a reliable witness. Go ahead.
That's incredibly reassuring. Okay, so we've covered the simulation, the VKO, we've covered the translation, the cross-species part. Now I want to get to the third pillar, which I think is the most visually beautiful part of this whole thing, trajectory inference.
This is where we truly unlock the dimension of time.
We mentioned earlier that development is a movie. Trajectory inference is how we reconstruct the plot of that movie. How does a stem cell decide to become a neuron?
TEDD 2.0 uses an engine called PAGE partition-based graph abstraction.
Which, again, sounds like a terrifying math concept.
The math is complex, but the output is intuitive. Think of it like a subway map. PAGE groups similar cells into stations clusters. Then it draws the tracks between them based on how the cells are changing over time.
And this introduces the concept of pseudo-time. I want to clarify this because it's not the same as looking at a clock.
No, it's not. Real time is day 1, day 2. Pseudo-time is progress. Think of it like an assembly line for a car.
Okay.
You might have a car that has been on the line for 5 hours but is only halfway done because a machine broke. You have another car that's been on for 3 hours but is finished. Pseudotime measures how finished the car is regardless of how long it actually took.
So it sorts the cells by their biological maturity.
Exactly. And the case study in the paper, the mouse fetal cortex, illustrates this perfectly. The cortex, it builds itself in layers, basically inside out. The deep layers form first, then the upper layers.
So you have the parents, the intermediate progenitor cells, or IP. They give birth to the deep layer neurons, DLN, and then later they produce the upper layer neurons, ULN.
Correct. And when they ran the PAGE analysis, the graph showed exactly that. A thick line starting at the progenitor cluster, moving it through the deep layer cluster, and then a separate branch extending to the upper layer cluster. It reconstructed the lineage perfectly.
But drawing the line isn't enough. We need the street science. We need to know what makes a deep layer neuron different from an upper layer one.
That's the marker gene analysis. The system runs statistical tests like Rancor and students' t-test to find which genes are unique to each stop on the subway line.
And for the deep layer, it found a gene called SOX5.
SOX5 is the ID card. It's a transcription factor that essentially tells the neuron, you are going to the deep layer. The system also found Anphia and Neurod2.
And for the upper layer?
It identified GRIA2, which is a glutamate receptor, and NCAM1.
So why does this matter? Why should a listener care about SOX5 versus GRIA2?
Because in many developmental disorders like autism, schizophrenia, or epilepsy, the layering of the brain is disrupted. Cells end up in the wrong place. By identifying these marker genes and placing them on that timeline, TEDD 2.0 gives us diagnostic targets. We can now look at SOX5 and ask, did this gene fail at pseudotime point X?
It gives us the mechanics manual for the brain.
It does. It moves us from just seeing the error to understanding the mechanism of the error.
It's a lot. We have a simulator, translator, and a time machine all in one dashboard. But I have to ask the difficult question. Is it perfect? What's the catch?
It's an incredible tool, but it does have one major blind spot for now. Currently, TEDD 2.0 only contains wild-type data.
Meaning healthy tissue.
Right. It doesn't yet include disease tissues like a tumor sample or a brain with advanced Alzheimer's.
So when we simulated the TREM2 knockout earlier, we were simulating the start of the problem in a healthy cell. We weren't comparing it to a cell that was already sick.
Correct. We are inferring the pathology from the healthy rules. That is powerful, but it's not the whole picture. To really understand a disease, you eventually need to map the sick network too.
And I assume the creators know this.
Oh, they do. The paper outlines the vision for TEDD 3.0 and beyond. They plan to incorporate non-wild-type data disease models. and they want to integrate bulk RNA-seq.
Which is the kind of data doctors actually deal with in clinics.
Exactly. Single cell is expensive and still mostly a research tool. Bulk sequencing is common. If they can bridge that gap, a doctor could potentially upload a patient's generic genetic profile, map it onto the TEDD trajectory, and see exactly where their development deviated.
And finally, they mentioned linking it to genomics.
Yes, connecting specific DNA mutations to these expression patterns. That would link the cause, the typo in the DNA, to the effect, the change in the cell's life story.
So let's bring this all back home. We started by saying we wanted to turn snapshots into a movie. But hearing you explain all this feels like we aren't just watching the movie anymore. We're starting to get our hands on the editing software.
That is a profound way to put it. TEDD 2.0 is what we call a generative tool. It doesn't just store answers, it helps you ask better questions. It creates new hypotheses.
Right.
Our researcher can now say, I wonder what happens if I delete this gene, and get a statistically backed answer in minutes, not months.
It fundamentally changes the speed of discovery, which raises a provocative question for everyone listening. If we can virtually simulate a gene knockout with, 89 significant gene changes and get it mostly right, are we approaching an era where we can solve a disease mechanism entirely in the cloud before we ever test a single subject?
We aren't there yet. Biology is still messier than our best algorithms, but tools like TEDD 2.0 are certainly building the runway for that future.
It's a fascinating, slightly sci-fi reality we're moving into. I want to ask you, the listener, which feature would you play with first? Would you jump into the time-traveling trajectory map to see how a heart cell is born?
Or would you grab the virtual eraser and start knocking out genes to see what breaks?
I'd highly recommend checking out the show notes. We've linked the paper and specifically look for the PAGE plots and those species clusters.
Oh, absolutely. Seeing the visual data really makes the time element click in a way that audio just can't fully capture.
It really does. Go check those out. It's been a journey through time, space, and species today. Thanks for diving in with us.
Always a pleasure to explore the data.
We'll catch you on the next deep dive.